Hello students! Today it's Biology 101 Lecture 4 and we are going to look at a really cool compound called carbon. Carbon is the backbone of life, and that is what we think is the most important element in organisms. Living organisms, as you know, consist mostly of carbon-based compounds. That is life as we know it. Life as we don't know it, well, we wouldn't know it, would we? But we would theorize that perhaps if it's similar to us, maybe not carbon, maybe something else similar, down on the periodic table, right directly underneath carbon, like silicon. But then again, that would be the life that we wouldn't recognize. All life that we do recognize is carbon-based. All the proteins, the DNA, the carbohydrates, and so on are carbon-based molecules. Carbon also happens to be the fourth most abundant element in the universe. As we all know, the most abundant element in the universe is hydrogen. But carbon is right up there. And, as we all also know, that if we look at the human body and we check out which are the elements that are most common there, carbon is the second most abundant element by mass. The first most abundant element by mass in the human body is oxygen. And this is that little cartoon with, actually it's sort of like a plastic, jiggly sort of bottle. And you could fill it up with some blue liquid, and that would come all the way up to the hips. That's 65% of a human body that is full of just oxygen. And then 18%, which is the second most abundant element, would be carbon. And followed quickly by that with hydrogen at 10%. And then nitrogen is only 3%, and the others are much less than that. We also know that carbon exists not just in the human body, but also in the biosphere. And when we look at the biosphere, there is an entire carbon cycle. So we can start anywhere we want in the biosphere and check out the carbon. Let's start with the atmosphere, for instance. In the atmosphere, carbon is only present as a trace gas. So if you notice the numbers, they're only 750 gigatons. So not a lot of storage in the atmosphere. But where is carbon mostly stored? It is mostly stored in the deep ocean. If you look all the way to the bottom, it actually shows you that the deep ocean has 38,000 gigatons. So while the atmosphere only has 750 gigatons, and the deep ocean has a giant reservoir of carbon. And as you can see, it moves from the oceans, from the sediments in the oceans. It comes out by volcanic, buried submarine volcanoes, and it circulates around in organisms that are aquatic, and then they also go into terrestrial organisms. We actually have plants which also store a lot of carbon, and when they have died and petrified, we use the half fermented plants as peat, fossil fuels, and then they respire, carbon dioxide goes in and comes back out. So it's an entire cycle where carbon moves all the way through the atmosphere and back. And what we would like to know is, how come is carbon so important? How come it is a molecule? How come it is an atom that is present everywhere? Why? Why does it have these properties? And the reason we'd like to know that is because if we knew that, then we would know what to look for in other life forms. We would know, this isn't there, so, we wouldn't be able to conduct life as we know it, or chemistry as we know it, in such an environment. But carbon is unparalleled in its ability to form really big and varied molecules. And it can also just bond with itself again and again and again repeatedly. We've even seen in space 60 carbons joined to each other as buckyballs. Carbon-carbon bonds are covalent bonds. They're very, very strong and stable, and they allow carbon to form an almost infinite number of other compounds. Carbon chains form the skeletons of most organic molecules, and these chains can vary in length and can vary in shape. They can be straight chains, they can be branching chains, they can be rings, they can have double bonds, they can have single bonds. There's an almost endless variation of carbon-carbon skeletons. In fact, there are more known carbon-containing compounds than all the compounds of the other chemical elements combined, except those of hydrogen. And because all organic compounds have carbon, most of them also contain hydrogen, which is kind of cool. This is a representation of a carbon molecule, a carbon atom. You see in the center this big black ball. Well, that is supposed to represent carbon. And it has 4 valencies, which are occupied by 4 other unknown at this moment other atoms. And what we like to do is, when we have a symmetrical molecule like carbon, we like to bond it with another symmetrical molecule like hydrogen. So we start out usually with a carbon compound with 1 carbon, 4 hydrogens, and that is actually methane. And then we start substituting each hydrogen for something else. And we come up with an infinite variety of carbon compounds. So, the carbon atom is tetravalent, and carbon is nonmetallic. And we know that the outermost shell has 4 electrons. So to get to the noble gas configuration of neon, it needs 4 more electrons. And so therefore, it just very easily makes covalent bonds. It's very happy to make them because there's an equal tug on the nucleus on all those electrons in that shell. When carbon is bonded to 4 other atoms, it acquires a tetrahedral shape. So if they're single covalent bonds, the molecule will have a tetrahedral shape. If there is a double bond present, then the molecule will no longer have a tetrahedral shape. It will look sort of like it's on the same plane. So, for instance, carbon dioxide, it does not look like, this is what carbon dioxide looks like. The atoms are in the same plane. They're not a tetrahedron. This is just a quick overview of all the valency electrons. So carbon is here in the periodic table. The first shell is full with 2, and the second shell has 4 electrons. And if we go down, directly down the periodic table in the next row, there is silicon. And silicon also has 1, 2, 3, and 4 electrons in its outermost shell. The innermost shell is complete with 2, and the second shell is also complete with 8. So silicon is somewhat similar to carbon, but not really, because it is a bigger, a more massive molecule, more massive atom, and has other properties associated with it then. This is just the tetrahedral structure of methane, represented in a 3D form. We are going to actually look at, or attempt to look at, these covalent bonds. Maybe we can look at them. I'm not sure if this will actually work, but let's see. It does work. Excellent.
Ionic bonding occurs between two atoms when one of the atoms has sufficient strength of attraction to remove an electron from the other atom. A sodium atom has one valence electron, with a stable energy level below containing 8 electrons. A chlorine atom has 7 valence electrons. If the atoms collide with sufficient energy, the chlorine atom will remove the electron from the sodium. The sodium atom loses its only valence electron and becomes a positively charged sodium ion. The energy level below now provides sodium with its stable octet. The extra electron completes a stable octet for chlorine, which becomes a negatively charged chloride ion. Covalent bonding occurs when neither atom has sufficient strength to completely remove the other atom's electrons. The atoms share electrons, and both atoms achieve a stable outer energy level. A hydrogen atom with one valence electron needs one additional electron to complete the first energy level. An oxygen atom has 6 valence electrons and needs 2 additional electrons to complete the second energy level. As the oxygen and hydrogen atoms collide, covalent bonding is accomplished by the sharing of electrons. By sharing an electron pair with the oxygen, the hydrogen completes the noble gas configuration of helium. By sharing with the hydrogen atom, the oxygen atom now has 7 electrons in its energy level, still one short of the stable octet condition. A stable octet for oxygen can be completed by sharing an electron with another hydrogen atom.
Excellent. Here is another one on bonds. And then we have this fun one, which is the chemical bond song.
Conviction, creativity.
First we'll start with ionic bonds. electrons for the nonmetal to receive. It all depends on what's needed to make the number 8 for the nonmetals our shell. And if it happens for them, they both become ions, charged atoms, they become ions. The metals positive, the non-metals negative, they become ions, oh yeah. The metals plus, the non-metals minus, and opposites they do attract. So what you get when they come together is an ionic bond. Sodium chloride. So what about those covalent bonds? It's not about loss or gain of electrons. Valence electrons, they are shared to complete the outer shells of the nonmetal sector bond. It's when nonmetals join to make covalent bonds with shared electrons. They're covalent bonds, not a transfer. Instead, they share valence electrons. Oh yeah, ionic bonds, covalent bonds, both of them chemical bonds. How are they made? What's the difference? Play the song again.
Or not. So here's another one. This is actually kind of cool because it's a cartoon of some tough guys.
So this video is a bit weird. I've got some new software I'm trying out. Now both of these big tough guys, they like to shake hands. And since they're both as strong as one another, the handshake's going to be right in the middle. Not to the left or to the right. It's going to be a symmetrical handshake. No one's pulling harder than the other. Now these two guys here, again, are sharing a symmetrical experience. Both of them are enjoy it as much as the other. So it's going to end up in the middle of their activity. But when you put them together, the big guy, well, he's so much stronger, he's going to pull the handshake over towards himself.
You are no match for me.
So let's look at the word covalent. Co means together. And valent, well, that means valence. And in this case, the valence electrons. Where are they? Well, hopefully you remember that they're the outer shell of an atom. Or maybe an ion. You can also think about copilot or cohabitation. So that's how you remember that covalent ones are the ones where electrons are shared. Now when these atoms share electrons, in some cases ions, they're going to have 2 or 8 electrons in the valence shell when they're done. This is stable. So if you're only looking at the first shell, that's going to have 2. And if you're looking at other shells, well, they're going to end up having 8 electrons once they've done their sharing to become stable. They'll look just like a noble gas. Now don't get this confused with, I'm listening to Bangarang, that's why everything's moving jerkily. Don't get this confused with ionic. Ionic is where electrons are transferred. We're not doing that, we're doing covalent. Co is sharing. So let me move these 2 atoms in closer together. And again, when they share their electrons, if they're sharing the first shell, they're going to end up with 2 electrons there, or if they're sharing any other shell, they're going to end up with 8 electrons together in those valence shells that I've indicated. And the covalent bond, well, that's going to be the shared electrons between them. So moving on, let's look at some examples. Now remember those 2 big guys, they both loved handshaking? That's like chlorine. They both really love electrons. So they're going to have a covalent bond, they're going to share the electrons because they both love electrons. How do I know that? Well, they're both non-metals. That's how you can work that out. The measure of electron love is electronegativity, which will come up a little bit later. Chlorine has a high electronegativity. Now hydrogen, that's like the little guy. Doesn't have a big electronegativity, only likes electrons a reasonable amount, a lot less than chlorine, but nevertheless the electrons will still be shared equally. If I put hydrogen and chlorine together, well, the chlorine loves electrons more than hydrogen, so it's going to pull those over, but it's still a covalent bond. Electrons are still being shared, if unequally, between those two atoms, the hydrogen and the chlorine. You can also get double bonds. This is ethene, you're going to need to learn that, and triple bonds. That is ethine. You'll have to learn that too. And strangely enough, they want you to look at these rather complicated structures here. Now, so far, we've seen an electron from each atom sharing to make the bond. But if both electrons come from one atom, that's called a dative covalent bond. And that's what I've drawn with the orange arrow there. That's the ammonium ion. The hydronium ion also. That top hydrogen, both electrons from the bond have come from the oxygen. The hydrogen is giving no electrons. Dative covalent bond. And the final one is carbon monoxide. In that triple bond there, the electrons in that top bond both come from the oxygen. They're not one from carbon and one from oxygen. So you call these a dative covalent bond, or you can also call it confusingly a coordinate covalent bond. When kids forget what a dative covalent bond is, I come over and pretend to strangle them. My two hands, the two electrons going out towards them. I'm not going to shake their hands. That would be a regular covalent bond. An electron from each. I'm going to go and strangle them. They will seem to remember that. Or if they don't remember it, they never mention it again.
That was too easy, mate.
That's funny. We have one more, and this one also shows covalent bonding.
Conviction, creativity.
Covalent bonding. Observe the representations of the various covalent bonds listed below.
However, this one, we're just going to keep that link because we're not going to go over it one more time. So we are done with, you can look at that link later, but we are just stressing covalent bonds that carbon makes with other nonmetals. And I just wanted to give you a good basis to understand, to remember what a covalent bond actually is. There is a whole branch of chemistry that studies carbon compounds, and that's called organic chemistry. The simplest form of an organic molecule is the hydrocarbon, and there's a huge family of hydrocarbons. Here is the carbon cycle with the formation of just the organic compounds. So what we see here is carbon dioxide in the atmosphere, and we also see carbon dioxide being taken up by organic compounds, the mushrooms, fungi, bugs that you see at the bottom. They're all using the carbon dioxide. Bacteria is giving off. Some of them are giving them off, and the bottom parts are actually taking them in. So we have a complete cycle of carbon. What was really interesting to try and figure out was, well, if we have carbon, can we actually synthesize complex biomolecules on their own by natural forces? And Miller-Urey did do an experiment to try and show the abiotic synthesis of organic molecules. They did a very simple experiment. They mimicked the second atmosphere of the Earth. So the first atmosphere of the Earth was just hydrogen, but the second atmosphere of the Earth was reducing, and it was mostly carbon dioxide, methane, ammonia, and stuff like that. And we are now on the third atmosphere, where it's mostly nitrogen and about 20% oxygen. So Miller-Urey actually found the components of the Earth's atmosphere billions of years ago, and they put together carbon dioxide, molecular nitrogen, hydrogen sulfide, sulfur dioxide, which they put in the atmosphere. And that was correct because at that time there was a lot of volcanic outgassing. And they ended up producing amino acids and sugars, like ribose, which is part of DNA, deoxyribose nucleic acid. And they did something very simple like this. So they simulated the ocean, and what they did was they made this nice round bottom flask, which they pretended had the ocean, and they proceeded to heat it. Of course, water will rise, it will become water vapor, and so it rose up this stem of the flask, and then they tried to simulate lightning. They pretended that it was going up in the atmosphere, and then in the atmosphere there are clouds, which are charged entities, and when they bump into each other, there's lightning, and lightning is an enormous amount of energy, and so that can produce high energy molecules. So something as simple as carbon dioxide and water can then become a very complex molecule because it's got this gigantic addition of energy. So then they rained all those molecules out into the condenser, and they checked them out and saw, well, what did we get? And what they got were simple amino acids, ribose sugars, which are pretty complex, and they were very pleased because then you could postulate that if you have the building blocks of life, then you could, under the right conditions, get those building blocks to become macromolecules, maybe even code information like DNA, and then be reproduced again and again and again, and form living organisms. So you have to start somewhere, and this is where it was a big breakthrough that you could actually start with the building blocks. So here we have, we're going back to carbon now, and what we're looking is the structure of methane, which I said was tetrahedral. It is the perfect symmetrical molecule, and we start substituting each hydrogen with something else. And when we do that, we always try and keep, let's see if I can get this pen to work. Maybe not. All right, no matter. We'll just plod along. And the second molecule you see in the middle of the slide is ethane. What we did here was we took away one hydrogen from this perfect molecule, which is let's see if I can get an arrow back. This perfect molecule, which is methane, we took away this hydrogen and instead put one of these on top of it. So we have the same molecule repeated on both sides. So we have 2 carbons and then 6 hydrogens, and that becomes ethane. It is no longer a tetrahedron, but you can see it becomes this very interesting shape. However, when we start putting in double bonds, like in ethene, we can have 2 hydrogens still, but we can take away 2 carbons still, but we can take away 2 hydrogens. And when we do that, this has to have a double bond because 1 hydrogen is missing, and then the molecule just comes into a single plane like this, which is kind of cool. A quick reminder that hydrogen, the valency is just 1. Oxygen, the valency is 2, so it needs 2 more electrons in the outer shell to get noble gas configuration. Nitrogen needs 3 more to have all the seats satisfied, and carbon needs 4. So these are the most common atoms that bond with carbon because they need either 1, 2, or 3. So the different molecules that carbon can form, it's almost unlimited. You can have a straight molecule, just continue on adding another carbon, another carbon, another carbon. Or you can start putting in double bonds, and then once you have double bonds, you have a different position Or you could have a branching molecule where you could just knock off one of these and push it up, and it becomes a branching molecule. It's no longer butane, but it becomes 2-methylpropane or isobutane. Or carbon will not bond in a straight line like this. It could actually bond as in a ring. And once you get a ring, then it has completely different properties. Cyclohexane, for instance, this is one. Every bond is satisfied. But benzene has 3 double bonds, and that's how we generally like to represent it. So hydrocarbons are organic molecules that only have carbon and hydrogen. Many organic molecules like fats have hydrocarbon components. So they're not all hydrocarbons, but they do have hydrocarbon components like a tail or something. Hydrocarbons can undergo reactions that release a lot of energy. We're going to look at an interesting property of these very symmetrical molecules. Once you have symmetry, you can tinker with it a little bit, and you can get astonishing models of new structures. And the molecular formula doesn't change. That's the cool part, because the molecular formula stays the same. But if you move in 3D space one of the bonds, you get a completely different molecule, and it has different properties, like different melting points, different boiling points, all sorts of stuff. So those things are called isomers. Isomers are compounds with the same formula but different structures and properties. And there's several kinds of isomers. There's a structural isomer, which has different covalent arrangements of their atoms. So just the covalent arrangement of their atoms is different. Or you can have a cis-trans isomer, which have the same covalent bonds, the exact same covalent bonds, but they just differ in spatial arrangements. They could be on one side or they could be on the other side. And then you have enantiomers, which are isomers that are mirror images of each other. So let's look at them one by one. So first of all, here's a summary picture. So structural isomers, if you look at them, this is exactly like that, but not quite, because it may have the same carbons, but here the 4 carbon are arranged in a different format, not in this kind of a jagged branch. A geometric isomer, these would be the cis and trans. So either the atom is on opposite sides or it is on the same side. And then you have enantiomers, and these are sort of like hands. So they are identical in every way except their mirror images. So they can never be superimposed. So if I took this one and I try to put it on this one, it wouldn't work. Just like our hands, they're mirror images of each other. We can't put them on top of each other because then if we do, then the thumb is not on top of the other thumb. So that is a mirror image or an enantiomer. Here are examples of structural isomers. So that would be where you start getting branches. So if we have pentane, 1, 2, 3, 4, 5 carbons. Again, 1, 2, 3, 4, 5 carbons, but not quite, because this 5th carbon is actually, the bond is somewhere else. And so instead of getting pentane and its properties, we get something else, which is called 2-methylbutane and has completely different properties. In the cis and trans isomers, cis means same side. So the whatever atom is here, it is here. It is, both of them are here. Or you could have a trans isomer where one is on one side and the other one is on the opposite side. Enantiomers, you have the mirror images. So this is a left hand and a left isomer, and that's a D, or a right isomer, a right-handed isomer. Life is very funny that way. Life prefers left-handed and right-handed molecules. So first of all, cis means this side. or the same side, and trans means the other side or the opposite side. So cis isomers have substitute groups in the same direction, whereas trans isomers have substitute groups in the opposite directions. And you might think this is a very small, nitpicky detail, but it's not. It endows different properties on these compounds. So chirality is another idea about symmetry. So when we talk about symmetry, we are obviously then immediately bringing in asymmetry, which means things are not symmetrical. Chirality is a property of asymmetry, and a molecule is chiral if it's not identical to its mirror image. That means it cannot be superimposed onto each other. So the chiral object and its mirror image molecule are called enantiomers. And it doesn't just apply to chemistry, it applies to objects too. For the best example of chirality is human hands. So for the left hand is a non-superimposable mirror image of the right hand. And no matter how you orient the two hands, it's impossible for all the major features to coincide. You can't shake the right hand of somebody using your left hand. It just doesn't work. And it's the result of, in humans, it's the unequal distribution of fine motor control. So it's really a different reason, but we end up with mirror images. Almost all organisms have only left-handed amino acids and right-handed sugars. So we know that life prefers left-handed amino acids, not the right-handed ones. The right-handed ones are preferentially not picked up. And the right-handed sugars, so the left-handed sugars, are not used. So this exclusive one-handedness means that all proteins and DNA are chiral. And chirality isn't just restricted to molecules in hands. It's everywhere. Let's take a look at this. So that was very interesting. Here is another one.
chiral object is one that is not superimposable on its mirror image. Your left hand is chiral. It cannot be superimposed on your right hand. Many molecules, including important drugs and natural products, are chiral. This is a ball and stick model of a carbon molecule with 4 different groups attached to the central carbon. For example, the 4 groups could be hydrogen, chlorine, methyl, and carboxylic acid. This molecule is chiral. That is, it is not superimposable on its mirror image. The 2 forms of a chiral molecule are called enantiomers. Using special rules, one form is labeled R, the other S. Enantiomers have mostly the same physical properties, but they behave differently in chiral environments. Molecules may contain more than one chiral center. For example, tartaric acid, which is found in many fruits and in wines, has 2 chiral centers.
So that was another very interesting way of looking at symmetry. Here is another example. If you've ever gone to the beach, you may have looked at shells. Most shells are coiled in the right-handed way. And this is a very common shell that we see on the beach. Flatfishes like flounder, oops, that's a typo, are left-eyed. These fishes like to sit at the bottom of water, at the very bottom of the ocean. And they lie there motionless and wait for the food to come by. And so they lie there for so long that one of their eyes actually travels around their head to the other side, so they have both eyes on the same side, so they can see better. And so flounders, for instance, are left-eyed, and halibut are right-eyed. We can also exploit this chirality by trying to diet and lose weight. So sugars are obviously needed to provide energy. But if we eat the mirror image of the sugar, if we don't eat the right image, then if we eat we would starve. We can only metabolize the right-handed sugar. Okay, so the left-handed sugar is just going to not be metabolized by us. We will still taste it as sweet, but we won't be able to metabolize it. And that's how we get 0 calories in artificial sweeteners. So here again, our enantiomers are left and right. Right is labeled as a D. And these are chiral molecules. They're non-superimposable on top of each other. They are mirror images, and usually only one isomer is biologically active. So as we can see now, even the tiniest difference in bonds will produce different effects. Properties of organic molecules generally depend on the carbon skeleton. The carbon skeleton is attached to hydrogen with covalent bonds, but if you start replacing the hydrogen with something else, you're going to start replacing it with different groups, and you get different results. So for instance, here is one nice molecule. It is, if you look at it carefully, so two 6-sided rings at the bottom. One of them actually has double bond, so that's a benzene, and then 2 that do not have double bonds, and then a pentane. And what you look at is on the left-hand side, it is labeled estradiol. So it's very similar to the molecule on the right-hand side, which is testosterone. The only difference you see is the first 2 molecules, if we start reading from the right-hand side, hydroxyl molecule present in both. Check. Methyl molecule present in both. Check. Same bond, same place. Then in testosterone, there is another methyl molecule which is absent in estradiol. And then in estradiol, we see there's a hydroxyl group which is absent in testosterone, and there's just one oxygen there, double bonded. So it's the testosterone is missing one hydrogen at the hydroxyl group level, of estradiol, and it is adding a methyl group, and that makes a difference between males and females. So estradiol and testosterone, these are so similar, and they're only differing in the chemical groups that are attached to the carbon skeleton. So functional groups are the components of organic molecules that are most commonly involved in chemical reactions, and that's why they're very important. The number and the arrangement of these groups give each molecule its unique properties, and there are 7 functional groups that are most important in the chemistry of life. For instance, hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, phosphate, and methyl. And we're going to look at each one separately. Well, here is a summary slide showing all of them. So you can see the hydroxyl represented in a ball and stick model. And its name is, the compound name is usually alcohol, or something all. And an example is ethanol. And you have the branching model with the carbons and all the hydrogens. And just one hydrogen has been knocked off over here and been replaced by an OH group, and that makes it an ethanol, which is an alcohol. Here we have a carbonyl group, which is a double bonded oxygen with a carbon, and you get ketones. So for instance, it's the same idea, okay, the same structure, but instead of the hydrogen here, which has been knocked off, and instead of the hydrogen here, which has been knocked off, you're replacing it with one oxygen with a double bond, that you get acetone. Yes, nail polish remover. And here you get propanol if you put the double bond here. So that's kind of cool because it's a completely different compound than acetone. And then you have the carbonyl group, which is 2 oxygens, 1 hydrogen, and 1 carbon. The oxygen is double bonded to the carbon, and the hydroxyl is bonded covalently to the carbon, and you end up with organic acids like acetic acid. You also get amino groups, and you get amines or amino acids from here. So here you have a nitrogen and 2 hydrogens, and you get glycine, for instance, which is present in space, and you get these molecules everywhere. They're very simple molecules. Then there's the sulfhydryl group, which is the addition of sulfur and hydrogen to get us thiols, and that becomes another amino acid, which is called cysteine. And of course, the phosphate group. The phosphate group is very important because it is part of ATP, or adenosine triphosphate, where we knock off one of these to get adenosine diphosphate and release a bunch of energy. So you can add it on to a glycerol and you get a glycerol phosphate. Or you can have a methyl group. You can add an entire methyl group to 1 carbon and you get a methylated compound. So instead of having 1 hydrogen, you can add another group of 1 carbon and 3 hydrogens. So here is a picture of the hydroxyl group all enlarged. And what it's really telling you is that it is a polar group because of the electronegativity of oxygen. And so it forms hydrogen bonds with water. The carbonyl group, they're sugars with ketone groups called ketoses. Those with aldehydes are called aldoses. Pardon me. And then we have the carboxyl group. That acts as an acid, an organic acid. And the amino groups act as bases. The sulfhydryl groups can cross-link. And so these are really cool because when we are going to talk about proteins, we'll see how they form this very nice stable bridge between 2 strands or 1 strand of a protein and make the beautiful folded structure of a protein. Phosphate groups, they contribute the negative charge. When they're attached, they confer the ability to react with water and to release energy. So that is how ATP is hydrolyzed and we end up with a bunch of energy. The methyl group actually is pretty important in the expression of genes. So it'll go and bond with our DNA, and it also changes the function of the sex hormones. ATP, let's talk about it. So one really important organic phosphate is adenosine triphosphate, which has an adenosine group attached to a string of 3 phosphate groups. And ATP stores the potential to react with water. And so it becomes ADP, and it releases a bunch of energy that is used by the cell for work. And this is what an ATP molecule would actually look like. So we have the adenosine group, which is right here. It's a nitrogenous base. And then we have 3 phosphate groups added on to each other. If we knock off one, so here we are with adenosine triphosphate, we add water to it, and you knock off one of these, so you end up with adenosine diphosphate and a bunch of energy. So all living things are made out of 4 classes of large biological molecules: carbohydrates, lipids, proteins, and nucleic acids. And we've already established that carbon is a very important component of all biomolecules. So macromolecules are large complex molecules, and the unique properties that we get from the complex molecules arises from the orderly arrangement of the atoms. Each cell has thousands and thousands of different macromolecules. Macromolecules vary among cells of an organism and vary more within a species and vary even more between species. There's a huge variety of polymers that can be built from a small set of monomers. And an example would be just like the alphabet. There's 26 letters in the alphabet, and yet we can end up with an encyclopedia or a dictionary with so many words that make sense. And we're not even talking about the words that don't make sense. So a lot of combinations can be made with a very small set of monomers. A polymer is a long molecule of repeating units of monomers, and there are 3 of the 4 classes of organic molecules. Carbohydrates, proteins, and nucleic acids actually have repeating units of monomers. Enzymes are specialized macromolecules that speed up chemical reactions, such as those that make or break down polymers. A dehydration reaction occurs when 2 monomers bond together and they lose a water molecule, hence dehydration. Polymers can be disassembled into monomers by hydrolysis, which is essentially reversing the idea of hydration. So on the top here, we see a dehydration reaction where we're synthesizing a polymer. We have a short polymer, any polymer. We're just going to call it 1, 2, 3, and we're going to add to it another monomer. We dehydrate, get rid of 1 water molecule, and you get a longer polymer. So now you have 1, 2, 3, 4 instead of just 1, 2, 3. You can break down a polymer by hydrolysis, and here you're going to add water to this long polymer 1, 2, 3, 4, and you can break that bond one more time, and you end up with 1, 2, 3 and a dissociated monomer. Carbohydrates include sugars and the polymers of sugars. The simplest carbohydrates are monosaccharides, or simple sugars. Carbohydrate macromolecules are polysaccharides, which are polymers composed of many monomers or many sugar building blocks. Monosaccharides have the molecular formula that's usually the multiple of 1 carbon, 2 hydrogen, and 1 oxygen ratio. For instance, glucose, that's the most common monosaccharide. Monosaccharides are grouped by the location of the carbonyl group, just as in aldose or ketoses, and the number of carbons in the actual skeleton. Okay, so for instance, we have on the left-hand side 3 aldoses, on the right-hand side 3 ketoses, and what you see is in the first, on the top, trioses or 3-carbon sugars, so that the formula would be C3H6O3, still the same formula, 1:2:1. And on the left-hand side, you get glyceraldehyde. And all we did was keep the double bond with oxygen at the very edge, the tip. Whereas if we move that double bond to the center of the molecule, we get a completely different compound called dihydroxyacetone. And if we go to pentoses, which are 5 carbon sugars, and they look like a pentagon, we have ribose on the left, and here again, the double bond of carbon and oxygen is at the tip. But if we move that double bond 1 carbon down, we end up with ribulose, which is not ribose, but something else. And then in the 6 carbon sugars, the hexoses, we have glucose, very simple molecule right here. The double bond with carbon and oxygen is at the tip. And in galactose, the double bond is still at the edge. But all we did was switch the position of the hydroxyl group. So you get something, glucose is present in fruits and vegetables, galactose is present in milk. So completely different sugars. And then we have fructose. Fructose is different from glucose because we moved the double bond from the tip carbon atom to the second one down the row. And you get fructose, which is cane sugar. And again, it's a different sugar. It's not the same. So this is a close-up look of the molecule called glyceraldehyde and a close-up look of the molecule called dihydroxyacetone. Note where the double bonded carbon with oxygen is. Same here with ribose and ribulose, and glucose, galactose, fructose. So although we usually draw the carbon backbone as a linear skeleton, in water, many sugars form rings, and they serve as a major fuel for cells and raw material for building molecules. So you can make proteins out of sugars, you can make sugars out of proteins. And so sugars are pretty important. Here is a linear form of a carbon compound, and then the same compound put together in a ring form. And so you get the same number of carbons, same formula, but now it's in a ring structure. A disaccharide is formed when a dehydration reaction joins 2 monosaccharides, and the covalent bond is called a glycosidic linkage. So here we have 2 molecules of glucose which are going to be dehydrated, and which means a water molecule is going to be taken out, and you're going to synthesize a disaccharide called maltose. Notice in the center there's a glycosidic linkage between carbon 1 and carbon 4. Here is at the bottom of the slide, you see a dehydration reaction in the synthesis of sucrose. You have 1 molecule of glucose, 1 of fructose, and you take away 1 molecule of water. You created a glycosidic linkage, and you end up with something called sucrose. Polysaccharides. Polysaccharides are interesting polymers of sugars. They're gigantic. They're big. They have many monomers, and they differ with not just the monomer type and how many, but the positions of the glycosidic linkages. So, for instance, starch, which is present in plants, it consists almost entirely of glucose monomers. And plants usually store the surplus starch as granules within chloroplasts and other plastids. And the simplest form of starch is called amylose. Here is a nice picture showing potatoes, and then inside the potatoes are storage structures called plastids. They contain starch granules in the potato cell, and if you actually opened one up, what you would see is an unbranched chain of amylose made out of glucose monomers. In the center, what you see is a picture of the person's arm, which has a muscle in it, and in the muscle is stored glycogen, which is reserve sugar, And when you look at the glycogen molecule, again, it is not branched, but it is made out of glucose monomers. And if you look at the very bottom part, there is a leaf, and inside the leaf there's you can see the cell itself, the cell wall, and it has green plastids in it. And if you actually looked at the cell wall, you would see this amazing structure with microfibrils of cellulose. But if you open up one of those microfibrils, what is it made of? It is made out of cellulose. So they're cellulose molecules. They're linked together by glycosidic linkages, but then there are hydrogen bonds linking the strands of cellulose together. So that's what makes it pretty strong. This is just a blow up of each part and showing you each part of the stored glucose. Glycogen is a storage polysaccharide in animals. It's stored mainly in the liver and the muscle cells. And when we hydrolyze glycogen, we release glucose when we need it. And this is a picture of the glycogen inside the muscle cells. Another close-up. Cellulose. So we're going to look at the tough wall of plants, and it's a polymer of glucose, but the glycosidic linkages are different, and the difference is based on the two rings of glucose. There's an alpha and a beta type ring. So if you look a little bit carefully, you'll see that the glycosidic linkages are actually not the same as what we saw previously. So here the linkages are all one side, and that's in glycogen. Here it's alternating. If you look at the glycosidic linkages, one goes up, one goes down. So it's different. And that is a close-up of the cellulose microfibrils. And this is a plant cell surrounded by a plant cell wall. And here is a nice picture of the structure of alpha glucose and beta glucose. So you'd want to know what is the difference. In the center is a structure just of glucose, general formula. But alpha glucose has the difference just lies in one OH group. It's either this side or that side. So when you have alpha and beta glucoses, you can make this kind of a ring structure as in a glucose monomer, or you can get a cellulose structure where the 1,4 linkages of beta glucose monomers are present. So alpha and beta glucose ring structures. This is a blow up of 1,4 linkage of a alpha glucose monomer and then 1,4 linkage of a beta glucose monomer. So we have starch, which is of alpha configuration, and it's largely helical. Cellulose molecules, which are of beta configuration, are straight and they're unbranched. Some hydroxyl groups on the monomers of cellulose can hydrogen bond with the hydroxyls of parallel cellulose molecules, and we saw that in a picture earlier. So enzymes that can digest starch, what they do is they hydrolyze the alpha linkages, but they cannot hydrolyze the beta linkages in cellulose. So when we eat plants, we cannot hydrolyze the beta linkages, and so the cellulose in our food passes through as insoluble fiber. But some microbes use enzymes to digest this cellulose. So for instance, cows and so on, they have a symbiotic relationship with these microbes, and since they have several stomachs, they can eat the food, and then it can just go and get digested in stomach 1, 2, 3, 4. So on. Chitin is another structural polysaccharide. It's found in the exoskeleton of bugs, crabs, shrimp, lobsters, caterpillars, and it's for structural support. And it also provides structural support for many fungi, the walls of the funguses. It is a derivative of glucose. You also find it actually on butterfly wing scales and the iridescent plumage of birds. So here's a picture. What you see here is this bug shedding its skin and emerging from its chitin exoskeleton so it can get bigger. And what you see in the structural formula up there is the structure of the chitin monomer. We also use chitin to make surgical thread, which is very nice. So here's the structure of the monomer. And what you see here on the right-hand side is the plasma membrane, the phospholipid bilayer. And what you see also are the chitin, the gray, thin boat-like, canoe-like slabs. And in between there are beta glucans. So these are not 1,4 linkages of glucose, but 1,3 linkages, and so you get a different kind of polymer. On top of all that, you get manoproteins. So they're proteins with sugars, and you get chitin right at the bottom of it. So that's what makes the exoskeleton pretty stiff. And what we have is, in this instance, a fungal cell wall with chitin.